Endomembrane system. See, endomembrane system. The endomembrane system consists of two endomembrane organelles. Two endomembrane organelles. Two endomembrane organelles. Those two endomembrane organelles. Two endomembrane organelles are, one is endomembrane bound organelles. Endomembrane bound organelles. Another one is non-endomembrane bound organelles. Non-endomembrane organelles. Those are here endomembrane bound organelles. Today we will see about endomembrane bound organelles. Tomorrow we will see about non-endomembrane bound organelles. Okay, so endomembrane bound organelles are endoplasmic reticulum. Endoplasmic reticulum. Endoplasmic reticulum. Golgi apparatus, lysosomes, and vacuoles. These are all called endomembrane system or endomembrane bound organelles, because in all these functions are coordinated with each other. Among endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles, the functions are coordinated with each other. Therefore, these are all called endomembrane organelles. Next, non-endomembrane organelles: mitochondria, chloroplast, peroxisomes. Those are called non-endomembrane organelles, because in between chloroplast, mitochondria, and peroxisomes, there is no coordination. That's why these are not considered as endomembrane bound organelles. Endomembrane organelles. Okay, so in between endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles, there is highly coordination. Coordination exists. So due to this coordination, these are called endomembrane organelles or endomembrane systems. Among those, first of all, we will discuss about endoplasmic reticulum. Endoplasmic reticulum. So presence of network or reticular structures are present in the cytoplasm near the nucleus is called endoplasmic reticulum. Endoplasmic reticulum. See here. In this one, this is eukaryotic cell. In the eukaryotic cell, there is a network or reticular tiny tubular structures. There is a network or reticular okay, presence of network or reticular. Reticular means what? Tubular, tiny structures present in the cytoplasm near the nucleus. These are all attached to the outer membrane of the nucleus. Those are called endoplasmic reticulum. So the endoplasmic reticulum, this endoplasmic reticulum divided into two structures. Intra-endoplasmic reticulum and extra-endoplasmic reticulum. Intra-endoplasmic reticulum and extra-endoplasmic reticulum. First one is the definition. Second point is endoplasmic reticulum is divided into endoplasmic reticulum divides the intracellular space into two types. So this is intracellular space. Intracellular space is divided into two types. One is intraluminal compartment. Another one is extraluminal compartment. Extra. See, this is intra. Extraluminal compartment. This is extraluminal compartment. Extraluminal compartment. This is intraluminal compartment. This is intraluminal compartment. This is called extraluminal compartment, which is located in the cytoplasm. That is called endoplasmic reticulum. So endoplasmic reticulum is divided into two types. Endoplasmic reticulum is divided into two types. One is rough endoplasmic reticulum. Another one is smooth endoplasmic reticulum. Endoplasmic reticulum. One is the rough endoplasmic reticulum. Another one is smooth endoplasmic reticulum. See here, rough endoplasmic reticulum. Rough endoplasmic reticulum. The ribosomes which are attached to the endoplasmic reticulum. Such type of reticulum is called rough endoplasmic reticulum. See here diagram I am showing. See, endoplasmic reticulum is attached to the ribosomes. Ribosomes are attached to the endoplasmic reticulum. Those are called rough endoplasmic reticulum. Rough endoplasmic reticulum. Rough endoplasmic reticulum is involved in the synthesis of proteins. Actually, you know very well, ribosomes are involved in the synthesis of proteins. So, when the ribosomes are attached to the endoplasmic reticulum, ultimately they are involved in the synthesis of proteins. Ribosomes are attached to the endoplasmic reticulum, and that type of ribosomes are called rough endoplasmic reticulum. So, the rough endoplasmic reticulum is actively involved in the synthesis of proteins and their secretion. That is rough endoplasmic reticulum. Smooth endoplasmic reticulum. So this side, smooth endoplasmic reticulum is involved in synthesis of lipids. What are smooth endoplasmic reticulum? First of all, we have to listen. So the ribosomes are not attached to the endoplasmic reticulum. Such a type of endoplasmic reticulum is called smooth endoplasmic reticulum, as it is involved in the synthesis of lipids. In animal cells, lipid-like steroidal hormones are synthesized by the smooth endoplasmic reticulum. Smooth endoplasmic reticulum In animal cells, smooth endoplasmic reticulum synthesizes lipid-like steroidal hormones and performs their functions in animal cells. Okay, so that is the explanation about the endoplasmic reticulum. Okay. Next, Golgi complex. Golgi apparatus. See here, Golgi, Golgi, Golgi is the first scientist that he observed dense light structure, dense colored, dense staining in the eukaryotic cells. Golgi was the first scientist that he observed thick colored structure near the nucleus in the cytoplasm is called Golgi complex. Golgi complex. Okay, near the nucleus, see here, that is like this. Like this. These are called Golgi complex. Okay, Golgi is the first scientist observed densely stained. Stained means colored. Densely colored structures near the nucleus. Hence, this name called Golgi body after the scientist Camillo Golgi. Before that, no one knows that Golgi complex is present in that eukaryotic cell. When he was introduced, since then, these apparatus used to call Golgi complex or Golgi body after the scientists who discovered Camillo Golgi in the eukaryotic cell cytoplasm. Okay, that's all. Next, they consist of flat, disc-shaped sacs. They consist of flat, disc-shaped sacs or cisternae. Cisternae means what? Here, the Golgi complex is covered by thin layer. This thin layer is called cisternae. Generally, Golgi complex, Golgi body are flat, disc-shaped or disc-shaped or sac-like structures are cisternae. Cisternae means what? These are covered by thin layer. Cisternae are 0.5 micrometer to 1.5 micrometer in diameter. Its length is 0.5 micrometer to 1.5 micrometer. 1.0 micrometer in diameter. Okay, that is the length of the Golgi complex. Okay, next here, these are stocked with each other. These are stocked one above the other. These are stocked with each other. These are stocked parallel to each other. Parallel to each other means what? So parallel. This is parallel to this. This is parallel to this. This is parallel to this. They are parallel to each other. These are stocked parallel to each other and consists of Two types of phases. Parallel to each other and contains two types of phases. Two types of phases. One is convex. One is concave. Convex form. It is also called forming phase. Forming phase. And another one is concave, trans phase. Trans phase, it is also called mature phase. Convex. Forming phase. This is forming phase. Concave. Concave. Trans are maturing phase. Here, the fixed phase, the trans phase, both are different. They are seems to be different. One is convex, one is concave. Convex, concave. It is the convex, concave, like this. Both are seems to be different, but are interconnected with each other. This is the trans phase. This is the trans phase. Mature phase. Okay, so this is next one is trans, nothing but mature phase. Both are different shapes, but are interconnected with each other and perform functions. That is the structure. This is the structure of Golgi apparatus. So don't get confusion. So this is convex. Convex. This is also called forming phase. Concave, trans, maturing phase. Okay. Functions of Golgi body. The major functions of Golgi body. The Golgi body performs various functions. The main functions of the Golgi body is packaging materials. The main functions of the Golgi complex is packaging materials. Materials to be delivered. Packaging materials to be delivered either into the intracellular cells intracellular cells are secreted out of the cells. See, packaging materials. Various kinds of food materials are packaged. So packaging materials to be delivered into the intracellular targets, means intracellular junction, intracellular materials. To be delivered into the intracellular materials are produced, are secreted out of the outside of the cells. For example, cellular targets means what? The packaging materials, they may delivered either to the inside of the cell or secreted outside of the cell. That is the major function of the Golgi complex. Materials to be packed in the form of vesicles. See, materials to be packed in the form of vesicles. Vesicles means small, minute structures. See, vesicles is here I will show you. These are called vesicles. Materials to be packed in the form of vesicles from the endoplasmic reticulum. Materials to be packed in the form of vesicles, from the endoplasmic reticulum and are fused to the endoplasmic reticulum and are modified in the Golgi complex before they are leading into the maturing phase. See, here proteins are synthesized. So those proteins are packed in the form of vesicles from the endoplasmic reticulum and are fused to the Golgi complex. When they are fused to the Golgi complex, they are modified before they are released into the maturing phase. So here, attach and leave the Golgi complex. In meanwhile, those are modified. Whatever may be the materials to be packed in the form of vesicles from the endoplasmic reticulum, those are fused to the Golgi complex and modified, matured before they are leaving maturing phase. That is the second function. Okay, so attach it to the fifth phase. Attach it to the fifth phase. Attach it to the fifth phase. And modified before they are released into the trans phase. Okay, that is the one function. Next one here. Next function here you will see here a number of proteins are synthesized. Here already discussed. Number of proteins are synthesized in the endoplasmic reticulum. See, number of proteins are synthesized in the endoplasmic reticulum. So those are all the proteins are those are all the proteins are synthesized from the endoplasmic reticulum. Those are modified in the endoplasmic. Those are modified in the cis phase and leave the trans phase. Okay, that is the third function. Fourth one is Golgi complex. Golgi complex is the major site for the synthesis of glycoproteins and glycolipids. Okay, so glycoproteins and glycolipids, these two are synthesized in the Golgi apparatus. Okay, that is the explanation about Golgi apparatus. Lysosomes. Here, lysosomes. These are membrane bound. Vesicular structures are formed during the packaging of materials in the Golgi apparatus. In the Golgi apparatus, materials to be packed in the form of vesicles. So when the materials are packing in the Golgi complex, at that moment, membrane bound vesicular structures. Those membrane bound vesicular structures are called lysosomes. See here, membrane bound. Like this way, membrane bound vesicular structures are called lysosomes. So these lysosomes are very rich in hydrolytic enzymes. These lysosomes, isolated lysosomal vesicles or lysosomes, both are same. These lysosomes are very rich in hydrolytic enzymes. Hydrolytic enzymes such as carboxylases, hydrolases, lipases, proteases, carbohydrates. Sorry, here carbohydrates. carbohydrates. These are all called hydrolytic enzymes. So lysosomes are very rich in hydrolytic enzymes such as carbohydrates, hydrolases, lipases, proteases. In these lysosomes number is more. And these are all the hydrolytic enzymes are very active at acidic pH. Lysosomal enzymes are active. Okay, these are all the enzymes are active at acidic pH and capable of digesting. These are capable of digesting carbohydrates, proteins, and nucleic acids. So the nucleic acids are two types: DNA and RNA. DNA and RNA. So DNA and RNA are collectively called nucleic acids. Those are digested by the hydrolytic enzymes. These are very active at acidic pH. Okay, that's all. This is the explanation about the lysosomes. Next one is vacuoles. Vacuoles. So the vacuoles are located in the eukaryotic plant cells. So the vacuoles are located in the eukaryotic plant cells of cytoplasmic material. Eukaryotic plant cells are cytoplasmic material. Plant cell. So this is the plant cell. In the plant cell, vacuoles are present. Okay, that is the first statement. The vacuoles are located in the eukaryotic plant cells of cytoplasmic material. That is one. Next one, generally, the vacuole consists of excess amount of water, sap, excretory materials, and other materials which are not required to the vacuole. So all materials are present. See, so all the materials are present inside of the vacuole. So this is the plant cell. In the plant cell, this is the vacuole. So the vacuole contains water. Sap means juice, like structure, excretory materials, and other materials which are not required to the plants. So generally, this vacuole is covered by single membrane. That single membrane is called chloroplast. The vacuole externally covered by a single membrane. That single membrane is called chloroplast. So this chloroplast acts as a selectively permeable membrane. It acts as a semi-permeable membrane. Okay, that's all. So about 90%, the vacuole can occupy about 90% of the plant cells. In the plant cells, excess amount of place occupied by the vacuoles. Actually, there are number of vacuoles are present. When plant cell is maturing, these are all the vacuoles are combined and forms a huge vacuole and pushes nucleus aside. That's why about 90% of the plant cell cytoplasm is occupied by the vacuole. This vacuole pushes nucleus to a side. Okay, that is the explanation. Generally, this chloroplast acts as a semi-permeable membrane. So this chloroplast facilitates transportation of various kinds of ions, molecules, materials from lower concentration to the higher concentration. See, from the lower concentration means from the surroundings to the higher concentration, from lower concentration to the higher concentration. From the surroundings, a concentration is lower. So from the lower concentration, ions, molecules, the materials are allows to enter inside of the vacuole. So that inside of the vacuole, ionic concentration is more than surroundings. Okay, so this chloroplast is selectively permeable to some molecules. So those molecules ions are transported from lower concentration to higher concentration. That is the explanation about chloroplast. Generally, in animal cells, the vacuole is absent, but some protozoans, some unicellular organisms. So unicellular cellular organisms consist of vacuole. That vacuole acts as a contractile. So in amoeba, the contractile vacuole helps in excretion. In amoeba, contractile vacuole acts as excretory. Excretory means what? Some waste materials are expelled out through vacuole. In some organisms, some unicellular organisms like protists. In the protists, see the food vacuole. In the protist organisms, food vacuole are formed by the engulfing the food particles. Engulfing the food particles. Engulfing the food particles. Engulfing means what? Swallowing the food particles. Engulfing the food particles. Swallowing the food particles. When it will swallow the food particles, in the protists, food vacuole are formed. Okay, that is the explanation about the vacuole. So these are all coordinated with each other. Endoplasmic reticulum, Golgi complex, lysosomes, and the vacuoles are coordinated with each other. Hence, these are called endomembrane bound organelles. Endomembrane bound organelles. Okay.